Okay, so we're going to move on to section 11-3. You all have been doing just regular Punnett squares. That's called a monohybrid cross. That was a review from 8th grade. That should seem very easy. But I think you all realize that things don't work as tall and short. And either people come out tall or short, or green eyes or brown eyes. Well, no, there's all sorts of in-betweens. And that's what we're going to be taking a look at here. So go ahead and write the title down. Write down our page numbers. Let me explain you what this is all about, and we're going to get a little more complex with our Punnett squares from where we had been before. All right, so please write this down. We'll have a vocab quiz one of these days on this stuff, but what you've been doing is called a monohybrid cross. You know, mono means one. So these were genetic crosses or genetic matings in which we only studied how one trait was inherited. So we only studied plant height, or what color the pea pods were going to be, or something like that. It was very simple, but it doesn't work. It's not always this simple. With humans, there's 20,000 some traits, they believe. So to follow just one, it's good to learn from, and it doesn't always follow Mendel's rules. Mendel's rules were correct, but not everything works that simply as they do in pea plants. And you do not need to write this. This is what I was just saying. We only studied two alleles, like a tall allele and a short allele, with the dominant recessive. Well, things don't always work that way, and we're going to take a look at some exceptions to those rules. Here's our first exception. So you can go ahead and number this number 1, and this is called codominance. I want you to think about what the word co means. Co means two things. Like if a coach names co-captains of a team, that means there'll be two captains of that team. So codominance means two alleles could be dominant at the same time. And that's exactly what we have here. So please take a minute and write this down. Codominance occurs when both alleles are dominant and each allele's phenotype is expressed in the organism. Take a look at the pictures here. The dog in the lower right is exhibiting codominance. It got one allele for brown eyes, another dominant allele for blue eyes, and each allele dominates in a different part of the dog's body. The brown eye allele dominates in the left eye, the blue eye allele in the right eye. Same thing with this chicken. It got a dominant allele for white colored feathers and another dominant allele for black colored feathers, and the different alleles dominate in different parts of the body. So we have two dominant alleles, that's where the co comes from, and they're both dominating just in different parts of the body. Now we're going to write this with different symbols because it's a different kind of rule to how we're going to show our Punnett squares. So here's how we're going to write these. We're going to write F for feathers with a superscript B. That's going to be the allele for black feathers in these chickens that we're going to practice with. So F for feathers with a superscript B for black, and then F for feather with a superscript W for white. Now these are just like big T, little t or something like that, except they're both dominant, and they're just a weird way of writing things. Okay, so this will be our practice in your notes. We're going to try to cross a chicken that has all black feathers with a chicken that has all white feathers. Remember, the only things you can choose from, these are the two pieces of DNA. You have an FB piece of DNA and an FW. So let's take a look on the next slide here. So here's our cross, so go ahead and write that down as your phenotypes. So we have one that has all black feathers with one that has all white feathers. Now I want you to think for a minute. All you can use is FBs and FWs. Which two would you use to make an all black feathered chicken? And which two would you make to make an all white feathered rooster? This is what you'd have to do. you would have to use one piece of DNA, this FB here, from the one parent, and it would have to have another one from its other parent. That's the only way it could have all black feathers. And then the one that's all white, its mom gave it this allele, and its dad gave it this white feathered allele. So go ahead and draw a Punnett square and set this up. Go ahead and draw your square. Put FB, FB across the two columns, and then put FW, FW to the left of each row. And go ahead and carry them into the Punnett square. And tell me what offspring you're getting. Tell me the probability of the phenotypes that you'll get, and the probability of each genotype happening. Okay, what you should have seen after you've done that is that they all come out FB, FW. So that would be some black feather, some white feather on the same bird. So you would write for genotypes FB, FW, 4 out of 4. That's 100% of the time, so that's a guarantee. And every single chicken from this cross would look black with white feathers. So they wouldn't be gray. It'd be some black feathers, some white feathers. So that'd be your phenotype. Black/white, 100%, or 4 out of 4. So that is codominance. Here's your second example. This one's called incomplete dominance. So this isn't going to be 2 colors on one creature or 2 phenotypes on one creature. This is going to be a little different. Here's our definition of incomplete dominance. This is when neither of the 2 alleles is dominant. So this is when neither of the 2 alleles is dominant. So if you get 2 different alleles, they actually blend together to make like a meet in the middle of the 2 phenotypes. I told you that before Mendel's day, all scientists thought that everything blended together. Like if your dad was tall and your mom was short, you were supposed to be medium height. Well, there was a little bit of truth to that, and that was called incomplete dominance. That's just not the way everything works. So here on this picture, we have some flowers that got all red alleles. We have some flowers that got all white alleles. The pink one's got 1 red allele and 1 white allele, and those 2 have mixed together to make the pink flowers. So of course we're going to have a different way of writing this, and let me show you that. We're going to use just a big R for red flowers and a big W for white flowers. Remember, neither one of these is dominant and neither one is recessive. So if we want to make red flowers, we'll put big R because they got a big R allele from the mom and a big R allele from the dad. So all they could make is red because that's all information they have is for redness. If we want to make a white one, then this got 1 white piece of DNA or 1 white allele from mom, 1 white allele from dad, and all this can make is whiteness. It has no other DNA to make anything else. And then here, if one of these was a heterozygous, remember hetero means 2 different, we got 1 for red DNA from the one parent, an allele for white DNA from the other, and this doesn't make red and white spots, it makes pink. These actually mix together to make pink flowers. So let's try this out so you have a good solid example in your notes. So I want you to cross a pink flower with a red flower and tell me the genotypes and phenotypes of the resulting offspring. So set it up like this. You have 1 pink parent and 1 white parent. And these would be your symbols. So these would be your genotypes. The pink one would have 1 red allele and 1 white allele, and they have mixed together to make the flower look pink. This parent has 1 white allele and another white allele. So go ahead and set up a Punnett square with those. Copy them into the boxes and tell me the probability of each genotype that you're seeing inside your boxes and the probability of each phenotype you're seeing inside of your boxes. All right, if you take a look at what you have, you should have 50% or 2 out of 4 should come out RW, RW. The other 50% or the other 2 out of 4 should come out WW and WW. So if you look at your phenotypes, what you should have is you have a 50% chance, or 2 out of 4, that the flowers will come out pink, and a 50% chance, or 2 out of 4, that they'll come out white. So that's incomplete dominance. Okay, this will be our third example, so there should be a number 3 up there. It doesn't always work so simply that there's only 2 alleles for a trait, like tall or short, red or pink. I mean, there could be lots of different alleles. And if that happens, then we call that a situation where there's multiple alleles. So this will be a definition for you. So the key word, the vocab word is multiple alleles. You can write down that definition. When a gene has more than 2 different alleles for a trait, you just stop right there at the comma. Now, if you have more than 2 different alleles for a trait, logically that means you can have more than 3 possible phenotypes. And we're going to see that as we go into one more example. Human blood type is a great example of this. And there are 3 alleles that make our blood types. One is a capital I with a superscript A. One's a capital I with a superscript B. And one's recessive, so let's draw that as a little I. If you look, we're writing these the same way that we wrote our codominance ones, because you're going to see that we see some codominance with our blood type. So, if you think for a minute, what are the different kinds of human blood? You can forget about the negatives and the positives. There's 4 main types. Someone could have A-type blood, they could have B-type blood, they could have AB, or they could have O. Now, there are 3 alleles that can combine to make these, but you need to remember each person only gets 2 alleles. You get one from mom and one from dad. But there are 3 that exist just out there in the population, but you still only get 2. Mom gives you 1, dad gives you 1. Let's take a look at the different ways these alleles can interact to make these different types of blood type. All right, if you take a look here with me, there's 2 ways to get A-type blood. So if you want to make this little side-by-side chart here, we'll have our 4 blood types going down. So to have A-type blood, you can get a dominant A and another dominant A, and that would give you A-type blood. Or one parent could have given you this codominant A allele and a recessive. Well, this A would dominate this recessive, and you would still come out with A-type blood. This is called being homozygous for A. This one's called being heterozygous for A. To have B-type blood, it's pretty much the exact same thing, but you have these B-type dominant alleles, or one of the B-type dominant alleles and a recessive allele. To have AB, we get some codominance here. Mom gave you maybe the A-type allele, and dad gave you the B-type allele. That means some of your blood cells are of the A-type, and some of your blood cells are of the B-type. And then finally, we can have O-type blood. O-type blood, you get 2 recessive alleles. Right, you don't get anything to make any kind of A or to make any kind of B, so you just get O-type blood. You can't make A-type blood or B-type blood, so you make O-type blood. Now, this is something you're going to have to memorize next time we have a test. You're going to have to know this chart. So if I say cross somebody with homozygous B-type blood with someone with AB-type blood, you have to be able to know this. So this is something you're going to have to be able to commit to memory. All right, and finally, we could have polygenic traits. Polygenic traits are traits whose genotype is controlled by 2 or more genes. Poly means many, so this is a many-gene trait. So not all of your traits are controlled by one set of genes. The way you could recognize these, we won't do Punnett squares for these, but the way you could recognize them is there's a lot of variation within a population for these traits. And here's your examples. Human height. We don't come out tall and short. There aren't just 2 human heights. No, I mean, if someone says they're 5'7" and someone else is 5'7", they're probably not even the exact same height, because one's 5'7 1/4", one's 5'7 5/8". So there's a lot of variability because height is actually controlled by 7 different genes. Okay, bone structure, muscle structure, all these other genes, growth factor, all control your height. Skin color is the same thing. 2 people might look like they have the same skin color, but you look a little closer and it's slightly different.